That's a picture of a platypus. That's our chapter opener. And the reason they showed that is because platypus has a bizarre sex determination program or sex determination mechanism. And there's a whole bunch of X chromosomes and Y chromosomes. It's pretty complicated, and we're not going to go into that. It's just an example that there are lots of different ways of sex determination. We're going to focus on the one we're most familiar with, the XX and XY system in humans, which is similar to what happens in fruit flies. And so, but there are a lot of other potential mechanisms. So there are several different mechanisms of sex determination. The one we're looking at is the X and Y chromosomes in humans or mammals or fruit flies. And I think we all know a little bit about this. If we have two X's, we're a female. And if we're an X and a Y, we're a male. All right, so that's sex determination. Thanks for tuning in. Turns out there's a little bit more to it. Well, we know this, so why do we have, so what do we mean by sex? So we have two sexes, males and females, and then, we've talked about meiosis, generating haploid gametes, and then fertilization and getting a diploid. The only reason I really have this in here is to rehash the sexual life cycle going between haploid and diploid. And so in order to do that, you need to have two sexes. In fertilization, there's a sperm fertilizing an egg. And so the sperm would have, well, I guess it would have either an X or it could have a Y. And the female would have just one X. So that's an either or situation for the sperm. And just again, there's other systems. There's the ZW system in birds. There's this XX and XO system in grasshoppers. But we're going to be looking at the XXXY system. And that gets complicated enough. We don't need to throw in additional sex determination mechanisms at this point. So males generate two types of gametes, X or Y. Females generate only gametes with an X chromosome. And so because the males can give two types of gametes, it's called heterogametic because it can give two different kinds of gametes with respect to the X chromosome. So males in humans and these XY systems, males are the heterogametic sex. In other systems, the female might be the heterogametic sex, and in females and humans, females are the homogametic sex, meaning that they make the same type of gametes, and males can make different types of gametes. And so then if you do a little Punnett square, you get an X and an X from the female, an X and a Y from the male, and when they come together, you can get 50-50 males to females. We'll talk a little bit about the X chromosome itself and a little bit about how the X chromosome and the Y chromosome determine sex, and then we'll start to look at some inheritance patterns. So these are what the chromosomes look like. The Y chromosome is small, and it doesn't really carry very many genes. The X chromosome is much larger and carries quite a few genes. So let's just put a gene on here. If we had a gene here, let's say the color blindness gene, there's no corresponding gene in the male. So that would look like this. And this individual would be colorblind because there is no allele on the Y chromosome to be dominant over the recessive colorblind allele. There's also this thing on the Y chromosome called the pseudo-autosomal region. So the pseudo-autosomal region, that means kind of like an autosome. So an autosome is a non-sex chromosome, and non-sex chromosomes have similar, when autosome pairs line up, homologous pairs, they have similar DNA sequence, which allows them to pair up during meiosis. And so there's a little bit of the X and Y chromosome that have similar sequence. So these little bits are homologous. So what that means is that they can pair up during meiosis, and they can even do crossing over and that sort of thing. And so there are some genes here, and if you have a gene here and a gene here, it'll be inherited in a more autosomal pattern. But if you have genes anywhere else on the X chromosome, it'll be an X-linked pattern, and there are some genes on the Y, and then you'll get a Y-linked pattern of inheritance there. So how does the heterogametic sex differ from the homogametic sex? Gametes of the heterogametic sex have different sex chromosomes. Gametes of homogametic sex have the same sex chromosome. And because they have different sex chromosomes, they can generate different kinds of gametes, and that's why they're called heterogametic. So we're going to talk about sex determination in humans and flies, and we know that in humans, it's the Y chromosome determines maleness. But in Drosophila, it's a little bit different. It's really just the number of X chromosomes. If you have 2 X chromosomes, you're female. If you have 1 X chromosome, you're male. And the Y chromosome doesn't determine sex. So, and you can actually get situations where you have just an X chromosome and no Y, and we call that an XO male, and it's still a male. And you can get an XXY female, and even though it has a Y chromosome, it's still a female because it's really the number of X chromosomes that determines sex in flies. But it's a little bit more complicated than that. It's not just the number of X chromosomes. So fruit flies have 4 pairs of chromosomes. So they have the sex chromosomes, and then they have chromosomes 1, 2, 3, and 4. So this is chromosome 1, and we also have chromosome 2, 3, and 4, 2, 3, and 4. So here we have 2 sets of autosomes and 2 X chromosomes. So we call that an X to A ratio of 2 to 2 or 1 to 1. So if we have an X to A ratio of 1, it's a female. But if we only have 1 X chromosome, we have an X to A ratio of 0.5, and that's a male. So it's just a little bit more complicated. It's not the Y chromosome that determines sex. It's the number of X chromosomes, and more specifically, the number of X chromosomes related to the number of autosomes. If you think of it in terms of the number of X chromosomes, that'll be fine for now. But this just gives you an example of the different kinds of things you would see. So a normal female, 2 X chromosomes, 2 sets of autosomes, X to A ratio of 1, you get a female. A normal male, 1 X chromosome, 2 sets of autosomes, an X to autosome ratio of 0.5, you get a male. This is also a male, XO, normal autosomes, 0.5 X to autosome ratio, so you get a male. If you have 2 X chromosomes and 2 autosomes, even though you have this Y, you're still a female. So that's a little bizarre. That's different than humans. We'll look at that in humans, and you might know a little bit about Turner syndrome and Klinefelter syndrome, and it's actually the opposite in humans. But here are some examples that show that you can manipulate the number of X's and the number of autosomes, and that will change the sex of the fly. So maybe we'll just do one example. So here we've got 2 X chromosomes, and now we have 3 sets of autosomes. So we somehow duplicated the number of chromosomes. So rather than having 3 extra pairs, now we have another extra pair on top of that. So instead of 6 chromosomes, there's 9 chromosomes. And that makes it so that the fly is sort of intermediate between male and female. It's an intersex. So let's just worry about the number of chromosomes. So know this information, because that'll come into play in a story a little further along. So what's the sexual phenotype of a fruit fly that has this kind of chromosome constitution. 2 X chromosomes, 2 sets of autosomes, so it's going to be a female. Doesn't matter, the Y chromosomes don't matter. Turns out that the Y chromosomes are needed for fertility for males. If you don't have a Y chromosome, then the males are sterile, but they still look like males. So that would be female just because it's just about the number of X chromosomes, not the Y chromosome. In humans, it's different. In humans, if you have 1 X chromosome, you're female. No matter how many X chromosomes you have, if you have a Y chromosome, you develop as a male. You can also get multiple X females. And so this is evidence that the Y chromosome determines sex in humans. So the Y chromosome determines maleness. And it turns out that there's a gene on the Y chromosome called the SRY gene, which is creatively called the sex-determining region of the Y. So you need that region of the Y chromosome to turn a developing fetus into a male. Here's a karyotype. So what's going on with this karyotype? We've got a bunch of chromosomes with their centromeres, and down here we see we have an X chromosome, but we only have 1 X chromosome and there's no Y chromosome. So that's an XO individual because there's no chromosome here. And would that be male or female? In humans, that's a female. And this is something called, what was capital T, but something called Turner syndrome. But it's definitely female. Here's another one, and this is what it looks like. So when you look at these karyotypes, you can actually look at these chromosomes, and that helps you. So this person would be Klinefelter syndrome, and there are some phenotypic characteristics that are characteristic of this particular syndrome, but sexually they develop as males, and that's because of the Y chromosome. And it's actually due to a little gene on there called the SRY that is present on the Y chromosome. So the X chromosome is important too. It contains genetic information essential for both sexes, so you need to have at least 1 copy of an X. This male determining gene, or the sex determining gene located on the Y, so even a single Y in the presence of several X's produces a male phenotype, and the absence of a Y results in a female phenotype, as long as there's an X chromosome. So there are some syndromes that go along with this sex-determining region of the Y. As long as you have that sex-determining region of the Y, you'll develop as a male. So if you look at the chromosomes and they look like there's 2 X's, you could get male development. And sometimes what happens is that sex-determining region of the Y gets moved over to the X chromosome. So you could have this SRY on the X, and you'll develop as a male. You can also have situations where somebody has an X and a Y chromosome. They have a normal SRY gene, so whatever that does, it's doing its thing. But that's just at the genetic level. To actually develop as a male, and this happens during development, is you need to express testosterone. And testosterone is a hormone, and a hormone needs a receptor. So if there's a hormone, in this case testosterone, here's a cell, you need to have some kind of a receptor to interact with this hormone. So this hormone will interact with the receptor, and then that will turn on genes and develop into a male. But if you don't have this receptor, then you can't respond to this hormone. Another word for testosterone is androgen. So there's other hormones that are similar to testosterone and grouped together, they're called androgens. So an androgen insensitivity syndrome is a syndrome where we don't have this receptor to respond to the presence of the hormone. And this SRY gene, normally on the Y chromosome, through a series of events, that leads to testosterone expression. Now, if you're really interested in that, which it's very interesting, take developmental biology. And you can learn all about how sex develops and how the different hormones work. And it's a very interesting class and subject. There's a picture of the Y chromosome, sex-determining region of the Y. Notice it's way up here. It's really close to this pseudoautosomal region, PAR, pseudoautosomal region. So this is the part of the chromosome that looks like the X chromosome. And sometimes you get pairing with the X and you get crossing over. And if you kind of mess up crossing over, you can pick up this sex-determining region of the Y. And that's how you sometimes end up with an X chromosome with that bit on the X chromosome. So that's sort of how the chromosomes determine sex, and there are certain inheritance patterns that are associated with sex. Now this is rare. This is a Y-linked pattern of inheritance, so this is in humans. And there are genes. So if there's a gene on the Y chromosome, I can't even think. I think there's a hairy ear trait that's Y-linked. And I don't know, all men seem to get hairy ears eventually, but there's some particular hairy ear gene, and it's on the Y chromosome. And so if this male has the hairy ear gene and the female has two X chromosomes, all these females, so circles are females in pedigrees, they have to get this X chromosome and one of these X chromosomes. These males get an X chromosome from mom, but they all have to get a Y chromosome from dad or they wouldn't be males. And there's only one place to get the Y chromosome, and so they all have an H, and so they'd all have hairy ears. So that's just an example of a sex-linked pattern of inheritance, but in particular a Y-linked pattern of inheritance. Now that's rare. But X-linked characteristics are much more common, and they were first discovered in Drosophila with the white eye gene, white eye mutant allele, and several other mutant alleles. And we can also see it in humans. Just to give you a little heads up, in humans, the way it works in humans is if females have a trait, they pass it on to other sons. If a male has it, usually a female will end up being a carrier, meaning she's heterozygous, and you can give her a little dot that tells you she's heterozygous, and then it'll show up in half of her sons. But as we'll see in a pedigree, if you see something where the trait is showing up in the males primarily, with females every now and then, start to think about X linkage. But we'll look at some better examples of that. But first, let's look at it in fruit flies. This is where it was discovered, and it was a very important discovery. It's probably right around 100 years ago, 105 years ago, this was discovered. This is a bit of a complicated figure, and we'll break it down. But the main thing here is what we're doing is a reciprocal cross. Reciprocal cross. Spelling that is always a challenge. Reciprocal cross means over here we're using a red-eyed female, and over here we're using a red-eyed male. So we call this crossing in a different direction. And the pattern of inheritance will depend. So we're really looking at this white-eyed mutant. If we start with a white-eyed male mutant, we'll get one pattern of inheritance. If we start out with a white-eyed female mutant, we'll get a different pattern of inheritance. So the key indicator of a sex-linked gene, one of the first ones, in this case we mean X-linked, is that reciprocal crosses give different results. If a gene is on an autosome, it doesn't matter if you use a male or a female in the cross, you'll still get the same result. So if you do the reciprocal cross, and this unfortunately the pen you can use with PowerPoint, the way I'm doing this with the recording, I can't really change much about the pen, so we're kind of stuck with it, which makes the writing look pretty crazy. But anyways, if we're starting with a red-eyed female, white-eyed male, the red-eyed female will give this wild-type X chromosome, and the male will give either an X with the white allele on it or a Y. But in either case, when these are fertilized, all of the F1 are wild-type. If you do the reciprocal recross and use a white-eyed female, you can get a X chromosome with the white allele on it from the female, and then either a wild-type X chromosome or a Y from the male. So then in the F1 generation, your females are all wild-type. They all have red eyes. It's because they have to get this X chromosome from dad here. And the males have to get the Y from dad, which means they have to get the white X chromosome from mom, and so they have white eyes. And the females got one X with a white and a plus. So a couple things are going on here. Notice that the results are different. If we do the cross in one direction, we get all wild-type F1. If we do the cross in the other direction, the reciprocal cross, we get half of our flies are wild-type and half of them are white. And not only that the white trait is linked with sex. Only males are getting the white-eyed mutation. So even if we did just this cross on the right, and we see this difference between males and females, that tells us that it's an X-linked trait, because males and females don't give you the same thing. So if we do a reciprocal cross, if we do both of them, we can tell that there's a difference right away in the first cross. If we do this cross on the right using a white-eyed female crossed to a wild-type male, we can also tell things right in the F1 generation. But if the only thing we do is this on the left, and we cross a red-eyed male to a red-eyed female to a white-eyed male, all of our F1 are wild-type eye color, but the female's heterozygous, and the male has an X plus and a Y. So now this female can give us either an X plus or an XW. The male can give us an X plus or a Y. When these things get together in fertilization, we get this ratio. We get 1/2 red females, 1/4 red males, and 1/4 white males. So that tells us that there's a difference between the males and the females. So the males, half of them are red and half of them are white. The females, all of the females are red. Over here, there's no difference between males and females. 1/2 of all the flies are red, and 1/2 of all the flies are white, and it doesn't matter if they're male or female. But in this cross on the right, we saw the difference up here. In the cross on the left, we saw that we didn't see the difference till the F2. So two things. If you see that the reciprocal cross gives you different results, and you see, or you see some kind of a difference either in the F1 or the F2 between the inheritance pattern of this gene whether it's a male or a female, then it's X-linked. If you did an autosomal cross with one gene, you would get, let's say, if red was autosomal, you'd get all red in the F1, regardless of sex, and you'd get a 3-to-1 ratio in the F2, regardless of sex, because that's an autosomal inheritance pattern. So there's a close-up of that. We've got our red-eyed female. She can give this gamete. The male can give this. If you have a white-eyed female, the female has to give the X chromosome with the white allele. The male can give a wild-type X chromosome and a Y. And this is sort of the big deal here, is that when this XW gets together with this Y, you get white male. And so that's an indicator of sex linkage. Then in the F1, so there's the parent generation, here's the results, where we had, in this case, all red. In this case, red females, but white males. So it looks like sex linkage is already here. This one we can't tell yet. We do the next cross. We take this heterozygous female, that's the F1 of that cross it to its brother, which sounds a little weird, but that's kind of the way you talk about it. And so we have a heterozygous female and a wild-type male. And then now, down here, is when we get this half red-eyed female, quarter red-eyed male, quarter white-eyed male. Here we had a difference between sexes in the F1, and then down in the F2, we don't see that difference again. So that's so that because the reciprocal crosses give different results, that means it's consistent with X-linked inheritance. This is looking at a colorblind male crossed to a normal female, and I would draw this like this: normal female, colorblind male, and the types of gametes we can get. So if this is a homozygous normal female and a colorblind male, then the only types of gametes we can get from mom are this. So all of the daughters would look like this, and all of the sons would look like this. So in the F1, we'd have both would be normal. And then if your brother and sister got together, who knows what would happen. All right, so then we take the colorblind female, normal male, gametes, F1. This is doing the cross in the other direction. So it's sort of like a reciprocal cross in flies, but we're using humans, so we'll do a pedigree. She's colorblind. He's normal. And then this is what are the things to look for in pedigrees. And these guys would now be carriers. And you get a little dot if you're a carrier. So that means we had to get this chromosome from mom, this chromosome from dad, chromosome from mom, chromosome from dad. Colorblind, carrier. But just looking at them, they have normal vision, the carriers. So this is what these X-linked things look like. Here's a female, has some kind of trait, normal male. All the females would be carriers, and all the males would have the trait. So an affected mom gives the trait to all of her sons. Concept check. Hemophilia is an X-linked recessive disease in humans. A woman with hemophilia mates with a normal male. What's the probability that their child will have hemophilia? And the probability is simply, we have a 1/2 probability. No, we are going to get so 1/2. So all of their sons will get it, but that's half their kids overall. All right. So what's the probability that they'll have a? Well, I guess that's good enough for that one. Y-link characteristics. We talked a little bit about that. We'll talk a little bit more about the Y chromosome. So here you have an autosomal pair. So you can get, so if this male determining gene somehow translocates or gets onto this is just showing the All right, a little bit more about X-linked chromosomes. So this is something called dosage compensation. Let me go back. So this is something called dosage compensation. And the idea is that there's a lot of genes on the X chromosome. And if males have one X chromosome and females have two X chromosomes, we'll have 2 times the gene product in females will be 2 times more. So the gene product will be there'll be 2 times as much gene product coming from the female as the male. But for most cells have to do the same thing, whether you're a male or a female. So you have to have the same amount of gene product, whether you're a male or a female. So we have to compensate for this fact that there's more of a dose. There's a double dose of X-linked genes in females, or you can think of it as a half dose in males. So dosage compensation is how do we sort of even that out? Well, what happens in humans, in mammals, is one of the X chromosomes in females. If you have 2 X chromosomes in a cell, one of those chromosomes is inactivated, and you end up with only 1 X chromosome in the cell. So during development, one of these X chromosomes gets inactivated, and we call that X inactivation. And what happens is the chromosome actually gets smushed down into a very tight little ball so that none of the genes can be expressed. This thing is called a bar body. It's also called the inactive X chromosome. And somewhere in here you still have an active X chromosome making proteins. But this one, the genes don't get expressed. The other thing that's interesting about that is if you have a cell and you have an X chromosome, sometimes that X will get inactivated, and then in another cell in the same organism, maybe the other chromosome gets inactivated. So X inactivation is random. And so what you end up with is an organism. That's an organism with different cells, a little square organism. And sometimes this one's inactivated, and sometimes the other one's inactivated. And so you sort of get this patchwork where sometimes one of the X's is inactivated and sometimes the other is inactivated. So we'll come back to that here in a minute. That's what happens in calico cats, is that in one of these patches, you have the reason that you have some sort of coat color gene on there. So that's kind of orangish. Let's say if you get a big O, you're orangish. And the other X chromosome is inactivated. Here's a patch that's black. And let's say if you get, we'll just put a different allele on there. We'll put the black allele. And if you get that allele and inactivate the other one, you get a black patch. And so most, that's kind of a cool pattern there. So this, in these guys, one X chromosome's inactivated. Over here, a different X chromosome's been inactivated. Same down here, one X chromosome inactivated, the other chromosome inactivated. So we call that genetic mosaicism. And this has to be a female cat, because this dosage compensation, this X inactivation thing, only happens in females. I did want to also point out the fact that this is called the Lyon hypothesis, named after Mary Lyon, and this inactivation process is called lyonization, and the inactive chromosome is called a Barr body, and that's named after, I don't remember her first name, but after another female scientist, Dr. Barr, and so that's dosage compensation. So that's the end of the chapter 4, the sex determination pathway, a little bit about dosage compensation, and go look at the problems to figure out, to get used to doing, looking at inheritance patterns with X chromosome genes, and I will try to put those problems online as well. So one thing I would add to the X inactivation story is certainly X inactivation occurs in humans. So one X will get inactivated, the other one will be active. The mechanism for how that works is really important, but we'll get into that later. It could be the other, so that when the left one got inactivated, it's possible for the right one to get inactivated, and then you've got your inactivated X. But we also mentioned that you could have 3 X chromosomes in humans, and that would be, so why isn't that bad? You can actually get multiple X chromosome syndromes in humans, and these individuals are relatively normal. If there's no Y chromosome, they would be females. But if dosage is important, and each of these X chromosomes is expressing genes, we'd have 4 times too much gene product. So gene products are RNA and proteins. But what happens is all of these X chromosomes, except for one, get inactivated. And again, it's random. And so that's why you can actually survive with multiple X chromosomes, and all of these X chromosomes will get inactivated except for one. Somehow the cell knows how to do that. If you have Klinefelter, one of those will get inactivated, and so you always just have one X chromosome that's active in any given cell in humans. So that was one thing I wanted to talk about. And then in flies, since we're talking about flies have the same kind of deal, right? We have one X chromosome we're male, 2 X chromosomes we're female. But rather than inactivating the X chromosome, so we have gene expression, and let's say we get down here and we make some proteins. And then in females, we have 2 X chromosomes. We have some gene expression, and we have some proteins. But in this case, I'm just drawing, this many proteins. Let's pretend there's 8. It'd probably be more like 8 million. And this has half as many. So one thing we could do is inactivate the X chromosome and then only express half of these proteins. But in flies, for whatever reason, the X chromosome is hypertranscribed. And that's just to let you know that there's different ways to do it. And so you have, it's the same net effect where we're evening out the expression between males and females, because if you have too much X chromosome gene product or too little, it's lethal and you die. And so you need to have the right amount. And there's two ways to do it. In humans and mammals, they get rid of one of the X chromosomes, and at least temporarily, and in flies, they double up the amount of transcription. And remember, transcription, DNA to RNA, and eventually we get down to protein, but that's translation. So the way we do this, we just do that more. All right, so that's dosage compensation in flies.